First I want to thank you all for being here, and of course thank the organizers for giving me a chance to talk. I'm a first-year postdoc at the National Institute of Standards and Technology, and my focus is on computational studies of sort of soft material systems specifically, and as I'll talk about on the next slides, even more specifically, semi-crystalline polymer systems. So I guess the imperative question to ask is what is a semi-crystalline polymer? And so semi-crystalline polymers are really one of the most common polymers we see in everyday life. I have a bunch of very easy examples I have up here. And I think another really telling example is if you list the first 6 recyclable polymers, those little symbols that you see on the bottom of your water bottles, 4 of those 6 are semi-crystalline in nature. And so this is really a very common type of material we see in everyday life. And the reason why it's interesting is that unlike a lot of other polymer systems, a lot of other homopolymer systems, they have a very interesting sort of dual microstructure. So we have, in this micrograph up in the corner, we have light regions which are dense and crystalline in nature, and we have dark regions which are amorphous. And we'll talk a little bit more about what that means, but the reason why we care about this is sort of that one of the fundamental ideas behind material science is that if we can control the microstructure of a material, we can control its macroscopic properties. And so in order to control the macroscopic properties of a semi-crystalline polymer, in order to design new semi-crystalline polymers for new and exciting applications, we need to be able to control their microstructure. And this is particularly frustrating in these systems because even very small changes to the chemistry, in this case comparing polyethylene to polypropylene, where we've just added one methyl group to every other carbon, these two semi-crystalline polymer systems could not be more different. And so in order to really understand how these chemical changes affect the macroscopic properties, we need to understand the microstructure. And this is where the next problem comes in. So looking at the concept of microstructure a little bit more deeply, semi-crystalline polymers are made up of two levels of microstructure. And so we have the crystalline microstructure, or the crystalline morphology as I like to call it. And in these sort of schematics we have, with varying chemistry, polymer chemistry, we can go through these different crystalline microstructures where we have bundles at low density, and as we increase the side chain count or the nature of the polymer we're putting into the material, we can go from bundles to sort of these chain folded structures to lamellae to almost single crystal structures. And each of these materials has vastly different macroscopic properties. But there's good news. We're really good at measuring these sorts of things. And so from an experimental point of view, we can come in and do scattering, we can do DSC, we can experimentally measure these semi-crystalline polymers and say something about their crystalline microstructure. That isn't so true of the connectivity between the crystals or the amorphous microstructure. And so here we characterize the amorphous morphology as being either tails, loops, or ties. So a tail chain is where we have a polymer chain that exits a crystal and terminates. We have loop chains which exit crystals and turn around and come back in. And we have tie chains which link separate crystals within the material. And what's particularly frustrating is if you look at the theoretical descriptions of these materials, whenever these amorphous morphology is what is most important in describing the mechanical properties. And so, but we can't measure it. There really isn't a lot of good solid metrologies that we can employ in order to understand in a real material what this amorphous microstructure is like. And so this is where we turn to simulations, and this is what I'm here to talk about. And so we can use simulations to predict and generate semi-crystalline microstructures, and then by doing thermophysical calculations on those semi-crystalline microstructures, we can compare things that we can measure in a lab to things that we can compute on a computer, and where those two concepts match up, we can say something about the microstructure, because we know in the computer what the microstructure is, even if we don't know what it is in the lab. So how are we going to do this? And so the scheme we came up with, and this isn't a particularly novel scheme, there are lots of schemes in the literature that look like this, is we create the microstructures separately, because one of the problems is if you try to simulate these crystalline microstructures directly, if you try to actually drive the system through the crystallization process, you end up with a much more homogeneous set of structures than you see in reality. And this is primarily due to the limitations of computer time and just the nature of the crystallization process. So what we do is we sort of break that process up. We do this unphysical generation of a semi-crystalline polymer system, and then we do some sort of calculation to test some material property that we can measure in a lab. So first, we generate the desired crystalline morphology manually. This is really just a geometry problem. There isn't any specific tools you need. You just need to sort of put the atoms in 3D Cartesian space and let them be. Then we grow in the amorphous morphology in such a way where we can control the extent of tie chains, loop chains, and tail chains in order to look at the effects of those on the last part where we do some sort of thermophysical calculation. And so steps 1 and step 3, super easy. Step 1, as I said, is just a geometric problem. And step 3, there are tons of tools out there to do these sort of molecular dynamics pooling experiments or thermophysical calculations that are very efficient and very well developed that make this part very easy. Step 2 is where I come in, because at this point in time when we were developing this project, there didn't exist, for the length scales that we wanted to study these systems on, a computational tool that allowed us to generate these semi-crystalline systems. And so we needed both a theoretical advancement in how we were going to generate these systems, and we needed a computational advancement because we needed to implement that theoretical advancement. And so the primary goals of my project over the past year have been to first design a molecular Monte Carlo scheme to construct customized crystalline and amorphous morphologies. This means that if an experimentalist looks through their microscope and sees a specific crystalline morphology, I can not only generate that crystalline morphology, but I can generate an ensemble of different connectivities between that morphology and see how those connectivities affect the mechanical or other thermophysical properties. And secondly, I wanted to implement this scheme in an open source package so other people could benefit. And perhaps most importantly, I needed to do this on kind of a short time frame. A 2-year postdoc is not something you want to waste on spending years developing a project. You need, and this was really the stopgap for the science I needed to do. So a continuing theme that I'm going to touch on is that one of the reasons I turned to Python for this project is because I needed it quickly. I didn't want to spend years developing a C++ project that I was just going to use for a few scientific projects. So briefly touching on, I'm not going to go into too much detail here, but just to mention, so the Monte Carlo scheme that we came up with, the sort of theoretical advancement, part 1 of the previous slide, is this semi-crystalline Monte Carlo. It's based on a couple, or actually many different works that are in the literature, but these 2 in particular. And essentially what we do is this is it's a regrowing scheme. It's a scheme which varies the topology within your simulation so that we can reconnect chains that used to be 2 tail chains, and we can regrow a portion of one tail chain and connect it to the other. And we do this in such a way where our statistical mechanics are, the statistical mechanics of the chain configurations are correct, and we do it in such a way that it's efficient. And even more important than both, perhaps even more important than both of those things, is we can do it in a controlled way. We can tune these biasing factors so that we can make it either more favorable or less favorable for the system to destroy tail chains and make tie chains, or vice versa. And so we can set these scaling factors at the beginning of a simulation and in turn control the relative propensity for forming tails, loops, and ties. And this really gives us what we need in order to generate not only controlled crystalline morphologies, but an ensemble of controlled amorphous morphologies.
Stand the mic for video purposes.
Yeah. Oh, sorry. And so, moving on to the code. So the code that we made is called TypeISIM, and so it's a Python, and I put molecular Monte Carlo in quotes for a reason, or in paren for a reason, but it's really a Python simulator. And so, first, why did I choose Python? And so if you're developing a simulator that requires performance, that is performance limited, you might not think of Python as your first choice because naive Python codes are very slow compared to naive codes in C++ or Fortran. But the reason why I chose Python was firstly for its rapid development cycle. As I said, I'm on a 2-year postdoc and I wanted this code as quickly as possible so I could move on with my science. I knew I had a rich ecosystem of tools I could rely on to help me get through this development process. And most importantly, one of my favorite sort of workflows that I use is Python plus Cython, where I write everything in Python. I look for the hotspots using some sort of profiler, and I drop those hotspots into Cython to get near C speeds where I need them. And so this allows me the flexibility and rapid development of using Python with, in some cases, the performance of writing in C or C++. And so sort of the philosophy behind the code base, and if you look in the code base, you're actually going to see these very directories, is as follows. And so we have our sort of core directory where we contain all the sort of containers and structures which describe the bonding and interactions within the system. We have engines. Engines are things that advance the system. This is either a Monte Carlo move or a Monte Carlo engine. Could be a molecular dynamics engine. I also have implemented a PACMOL engine for those of you who do MD simulations. And so that's sort of a spatial optimization engine that's really good for setting up initial configurations. But it's sort of an abstract idea of how we can take the initial system state and advance it in some prescribed way. We also have computes, which is where our thermophysical calculations lie. We have the potential directory where we describe how everything is interacting. I'm going to talk about molecule a little bit later because one of the sort of nuances of this specific simulation and one of the reasons why I think it was necessary for me to write a new code was I needed ways to sort of pick out certain pieces of the simulation and interact with them efficiently without having to sort of keep track of where, and without having to search through the system every time for specific chunks of the system. And geometry is just sort of helper functions. And so to kind of give it away ahead of time, what we did to make the simulation fast enough, and I say fast enough because I don't claim this to be the fastest MC code that's out there, but I claim essentially we were able to make it work for the system sizes that we needed. And we use NumPy arrays and vectorized operations wherever possible. As I said before, we write Cython plugins for the slow spots. I'm going to talk about our use of masked arrays for interacting with chunks of the system. And finally, this was something that came up several times during the development is, you shouldn't do more work than you have to. Sometimes if you think about the way you're doing a calculation, you can change it and optimize it and get much more speed up than you would by parallelizing it or doing some other complicated development. And so as you can see, TypeISim is a very small code base. It's very young. It's 8,500 lines and 14 Cython extensions. One of the sort of things I'm kind of proud of is that it only has 2 non-standard library dependencies, and that's just NumPy and Cython. And so in terms of installing it and getting it running, it's exceptionally simple. I also want to give a shout out to a number of other molecular dynamics codes that I drew inspiration from. I wouldn't have been able to develop this code if I hadn't have worked on or worked with these code bases in the past. So what does a TypeISim simulation look like? We're talking about sort of a complicated thermophysical or thermodynamic simulation. So what is the code that generates the simulation actually looks like? And so if you look, it's just a simple, really it comes down to, it's just a simple Python script. And this is really very similar to the way a number of other simulation packages work, in particular HumDBlue. And so we instantiate a system object. We describe the non-bonded interactions. We describe the bonded interactions. We set up a simulation box. So the way these simulations work is they're in sort of a confined periodic space. And so we need to say how big that space is. We describe our initial conditions. I just have placeholders here right now, and we add those initial positions to the system as a chain molecule. Finally, we create our Monte Carlo integrator. We add any number of Monte Carlo moves that we want to apply in the simulation. In this case, we're doing a fixed endpoint configurational bias Monte Carlo simulation, so we just have this one move that we apply, and finally we run it. And so in about 30 lines, you're able to construct and run a relatively complicated molecular Monte Carlo simulation that uses a pretty complex move set. And so this is all you would need to run that simulation. So going through some different pieces of the code in a little bit more detail, and so possibly the first or second most important part of this code base is the system object or the system class. And so early on, I was sort of going back and forth between representing everything in this way, where we have our Cartesian coordinates and our image flags and our types and all of that represented as simple 1D NumPy arrays, versus sort of going a whole nother level of abstraction and creating individual atom objects and atom classes for every bead or every atom in the simulation. But I decided that in terms of working with Cython and in terms of making the cache a little bit more happy, we would go with this sort of direct 1D array approach. The other benefit of going with this approach, this sort of less object-oriented approach, is that if we wanted to translate this to C++, it would actually be C++ or even a Cython extension. It would be exceptionally easy to write these as sort of C++ vectors or C++ arrays. When I was designing this class, the main ideas I was sort of keeping in mind is early on, I knew that I needed to be able to add and remove atoms and molecules very simply. And so, and when you look at a lot of other molecular dynamics codes or Monte Carlo codes, this isn't necessarily, less so for Monte Carlo codes, but this isn't necessarily the case. A lot of times you find that they use fixed length arrays and they're relatively reticent to allow you to add and remove beads. But because of the nature of the simulation that I was using, I needed to be able to do that. And so this system class is unit tested, and it's set up so that you can pretty relatively efficiently add and remove any number of beads or molecules you want for a specific given Monte Carlo move. And this allows you a lot of flexibility in terms of developing new Monte Carlo moves within TypeISIM developing new Monte Carlo moves. So I mentioned the sort of molecule views, and this is something that another thing I identified early on that we needed in order to make this code efficient is I wanted a way to take a subset of the system class and reference chunks of the system class efficiently. So the reason why we need this is in a simulation of, let's say, 1,000 or 2,000 or 10,000 polymer chains, we might need to take one polymer chain and shift all of its positions by a given amount. And so rather than creating a specific mask each time that Monte Carlo move came up and applying it to the system array, we wanted sort of an abstraction that allowed us to easily say, you know, this chain object corresponds to chain 1, and any effects that we apply to this chain object through these masked arrays sort of propagate directly into the system object. And so I guess I didn't mention the way we achieve this is through a NumPy feature called masked arrays. And so this allows us to sort of reference a NumPy array that already exists and apply a mask to it. And so now this self.x is sort of referencing a subset of system.x and allows you, if you're careful, to modify just the subset of system.x that you want to modify. There are some issues with masked array. It doesn't always do what you'd expect it to do. Sometimes when you operate on the masked array, it just operates on the whole underlying array instead of the mask part. But overall, if you're careful, this can be sort of an efficient way, both memory efficient and memory efficient way to sort of interact with your system without having to copy data around a whole lot. So in terms of the Monte Carlo moves themselves, this is again a case where I was going for ultimate simplicity. And so one of the other issues I found when I was considering other Monte Carlo packages was that a lot of times there was a complicated set of things you needed to sort of follow, a complicated set of rules you needed to follow when you were creating a new Monte Carlo move. And this often made it difficult to implement the sorts of Monte Carlo moves I needed in my specific simulation. And so in TypeISIM, the only things that a subclass of Monte Carlo move need to do is they need to implement an attempt function, and they need to, in the attempt function, they need to set three things: the accept flag, the abort flag, and the unew flag, which is just the potential energy of the new state if it was accepted. And so this allows us to do arbitrarily complex moves, or set up arbitrarily complex Monte Carlo moves within the code base without having to really worry about messing with a lot of the other machinery. You just have to basically write an attempt function which will have access to the system object and can modify that system object in whatever way is needed for that specific Monte Carlo move. So Yeah, okay, so the last part of the program that I want to talk about is sort of the pair interaction. So if you talk to any molecular simulator and you ask them what the slowest part of their code is, they're going to tell you, or they should tell you, it's the pair interactions. This is the probably the worst scaling part of any MC or MD codes, and it's because it basically scales as n squared with the number of particles in the system. And so this was an obvious place I knew I was going to have to tackle when I was writing this code base, and so we made this calculation efficient through sort of two methods. First, we use Cython, and so you'll notice that this code has types and doesn't look necessarily all that much like Python anymore, but it is. It's just written in a Cython extension, which compiles to C. And so this gives us near C speeds for this calculation, and furthermore, we can speed it up by using the OpenMP-based parallelization using this p-range loop. Finally, we implement a number of sort of pretty common computational algorithmic optimizations using neighbor lists, where we keep track of which beads are close to one another, and very specific potential energy calculation algorithms for very specific cases that reduce the amount of computation we need to do for certain types of Monte Carlo moves. And so just to kind of finish up quickly, what do we get out of all of this? We can actually generate, or we get what we were looking for. On the left of each of these sort of pairs, I have the crystalline morphology shown, and on the right is the amorphous morphology that I've grown into it. And the reason why these look like nice little cubes is because they're actually in periodic space, and so you can imagine tiling these infinitely in every direction. But we can see here that I'm able to generate semi-crystalline configurations for a pretty wide variety of crystalline morphologies. And the thing I'm not showing here is I can actually pretty tightly tune the degree of red, green, and blue monomers within the amorphous region, which tunes the degree of ties, loops, and tails, which was an important feature of this code base. And I won't harp on this at all because I'm running out of time, but essentially what we've been able to do is we've been able to take these configurations and plug them into molecular dynamic simulations and pull on them, measure their stress-strain properties as they deform, and what we're finding is we can get detectable sort of stress-strain features based on the number of tie chains in the material. And this is something that we literally can't measure in experiments, but is directly important to sort of fatigue properties and crack properties in these materials, and had some of our collaborators pretty excited. So to finish up, I just want to say that TypeISim is a very young, but sort of, I think, promising, simple molecular Monte Carlo tool. It's listed up here on the NIST GitHub. I have a number of other features and things I'm looking to do in the future, but for now, it's doing what I need it to do. So thank you.
Very much. Got a couple of minutes for questions?
Just curious, how do you validate your results?
So the structures that I generate. So it's pretty difficult, but essentially now, when we talk to our collaborator, he gets, so even though they can't measure the amorphous region directly, they can, based on theories, they think they know what's happening in terms of how the number of tie chains are changing. And he says he sees trends just like, we have a collaborator who sees trends very similar to what we're seeing here, but we're sort of testing this effect directly, whereas they're testing it sort of indirectly using some theories. And so we validate by comparison to experimental data as best we can.
I mean, when you say you test it directly from the model, or you actually test the experiment?
So in the experiments, what they do is they make a chemical change to the polymer, and they think they know how that chemical change changes the number of tie chains. And then they test the mechanics of those two systems, and we see similar changes when we sort of replicate what they think is going on and to their experimental data.